pleasure to introduce you to Dr. Nikolaos Daskalakis this afternoon. Nikos has an impressive CV. He received his MD from the University of Athens in Greece and went on to pursue his PhD in neuropsychopharmacology at Leiden University, where he also completed a postdoc in neuropsychopharmacology and clinical neuroendocrinology. He then joined the Icahn School of Medicine for a second postdoc in Systems Biology and joined the faculty in 2016.
Glad somebody brought their stethoscope.
Yes.
Okay. So Nikos moved to Boston in 2017 and is currently an associate professor of psychiatry at Harvard Medical School and an associate member of the Broad Institute and the MIT Computer Science and Artificial Intelligence Lab and the National Center for PTSD. In addition, he is also the director of the Neurogenomics and Translational Bioinformatics Lab at the McLean Hospital, where he conducts research focused on the connection between stress and brain functions using a wide range of functional genomic approaches. He has a very active lab and attracts grad students and postdocs from all over Europe and the US, despite the cold Boston winters. I started collaborating with Nikos many years ago in the context of the Psychiatric Genomics Consortium, where he currently leads the Systems Biology Working Group for PTSD. His work has a large impact in post-GWAS approaches to gain a deeper understanding of the individual differences in the expression of symptoms in complex neuropsychiatric traits such as PTSD. And I'm very excited to be able to announce that Nikos just received an R01 to follow up on our latest PGC-PTSD GWAS findings. Most recently, Nikos has published a paper in Science, and he will tell us more about this work in his talk entitled Systems Biology Dissection of Trauma-Related Risk and Disease Processes Across Brain Regions, Omic Layers, Cell Types, and Blood. Go ahead.
Thank you, everybody, for being here and in the Zoom. I will share and also stop the camera. Is everything okay in the Zoom side?
Yes, looks great.
Yep.
Mute myself. I want to stop the camera. Okay. Okay. So the title of my talk is very similar to the title of the paper, which is the main part of my talk, but I will give a background and some connections to my future work. So an easy way of starting is talk about the stress response. And as you know, all mammals, including humans, have a hardwired system how to respond that is very conserved and help them to, when they encounter a challenge, here a tiger in the picture, how to do this immediate reaction they have to behavioral do, the fight, flight, or freeze response. And largely the hardwired part of this involves the autonomic part of the stress response. But when the threat is gone, they need to shut down this initial reaction with systems like the HP axis and then product of cortisol to basically return to homeostasis. But something happens in individuals that have stress-related disorders, and these are not only mental disorders. Something happens in this second phase, the return of homeostasis doesn't happen. So a goal in the line of work I do, and I think is an important thing, is to understand these individual differences in the stress response as a basis of understanding not only stress-related disorders. I believe all mental disorders have that core thing they need to address. On the left side, you can see an example from schizophrenia, the classic figure reprinted many times from textbooks, that in schizophrenia, more genetically related you are to schizophrenic, the higher the probability you have schizophrenia. But that graph goes, in the case of monozygotic twins, only up to 48%. So what about the environment in this situation? On the right side, you have a disorder, PTSD, that by definition, according to DSM, to meet this diagnosis, stress exposure has to be part of what the psychiatrist assesses. But nevertheless, all those stressors that are very severe and traumatic are not infrequent. But the probability of having PTSD as an outcome to those exposures is not, the minority of the people get PTSD as an outcome of those bad experiences. So to give you the dipole of psychiatry, that where environment is influential and understanding individual differences in both extremes. A typical way of going after this, and it's a bit simplistic, is to do a psychiatric phenotype GWAS, genome-wide association study, in which you are trying to accomplish the statistical connection of genotypes that are in the order of millions of letters of DNA that can be binarized to connect it to a phenotype that in most of these studies is very simplistic, yes or no, having or not having a lifetime diagnosis of that disorder. Because of the simplicity of this analysis and the large scale amount of tests you do, statistically, to gain power to identify, safely identify loci, the so-called GWAS loci, you need high numbers of cases in this analysis. And in the middle, you see that in schizophrenia, to arrive to this exponential growth of genome-wide association loci in relation to sample size, this can happen, but you can have other disorders, like in this MDD case, that you don't get so easily an investment return to case numbers, possibly because of various issues of heterogeneity or different levels of irritability and environmental influences. PTSD, luckily, with the work analysis led by Caroline and Adam and the entire PTSD consortium, PTSD consortium, PTSD is somewhere in the middle, where right now, when the total sample size is approximately 1 million, we have a return of investment and have now 100 loci associated with PTSD. But what comes next is how to interpret this loci. And system biology is a path of not only gaining power in statistically speaking, but also getting biological understanding of where these loci are, what type of biology they affect, what are the brain regions, what are the developmental periods, what are the cell types. And this relies on the fact that genetics, the most proximal outcome that they affect is molecular biology. They don't directly affect the phenotype. There are many steps until then. So the system biology premise is that you can introduce the relationship that genotypes have to molecular networks and how molecular networks can explain more complicated neurobiological phenotypes until you understand the categorical clinical outcomes or the continuous scale. Here is a test case to give you an example of how this can be done. On the left side, you can see the catalog of human tissue that are in human tissues and especially brain regions and cell types and developmental periods that are now being sampled across the globe in large scale, where the funding agencies have done a significant investment of creating reference data sets where deep biology is measured, but at the same time, as the minimum, genotype is available for those samples. So the idea is that if you get the data of these reference panels, in the most simplistic way of thinking, you can create an equation to link the genotype with the quantitation of molecular biology that in this example is all about gene expression. And gene expression can be at the level of gene, it can be measured at the level of splice variants, it can be circular components of RNA, it can be cell type specific. But depending on the disease and the question of interest, you can focus on the biology you care about. Let's say inhibitory neurons in amygdalas being most relevant for PTSD, or fetal brain in schizophrenia and autism. But when you do that, in this particular case, Heike, a student in the lab, has selected DLPFC as her target tissue. She got access to the DLPFC reference panel created by the phase 1 of CycleNCode Consortium, and she trained models of gene expression, plain gene expression. And she compared her data with another reference panel from GDEX. In the end of the day, you can judge those models as machine learning experts do. Are they predicting good variants? Can they validate? Can you go to another tissue and actually confirm if they work? But the good thing is that machine learning framework has an understanding of classifying models in relation to how good they are and what they are predicting. But at the same time, while you're waiting for these models to get better and better, you can go to big data sets and actually apply them and see what conclusions you can make on the topic of interest. One scenario is on the left side, a health system or a biobank in which you have individuals that you primarily know only their genotype, and you have at the same time a lot of phenotypes. And those phenotypes could be how much alcohol they drink, it can be what is the exact centimeters of their height, it could be continuous categorical variables you know for them in detail. But biologically, you know only the genotype. If you use those models, you can start seeing them with a different lens and start imputing some biological information out of them and start doing association testing. Another way of doing it is you can go to other people's work that have done already the association testing of SNP to phenotype and just convert their result to your own result that is focused on a certain biology. So with this framework, you basically convert the risk SNPs, so SNPs that predict diagnosis, to genes that predict diagnosis in a particular tissue, context, and you name it. But the most important thing is that these are, again, risk genes. They might have the same problem that GWAS has in relation to how much variance they explain, but you have a very testable hypothesis. You know the tissue, you know the context, you know potentially the drug. So you can do actually an experiment or do a biomarker study to actually validate all those inferences. We did this for the first time in the phase 2 of PTSD GWAS. The GWAS from Caroline's paper in 2019. And back then, the power, in terms of power of GWAS and power of models, got us only 2 genes, one in the blood and one in the brain. Nevertheless, we went into doing validation work, also collaborating with the Marine Resilience Study. We also, not only we wanted to see if genetically predicted expression effects are correlating with the ones that you really measure in samples, but we want to also see if genes that are predicted to be related to PTSD, do they also change as a function of exposure itself? So in the 2 genes that we had, both of them were different at the pre-deployment study in this study in the direction that their genetics predict. But as a function of deployment stressor, they changed, one in the same direction as the susceptibility had, and the other had a compensatory change. We also start developing the logic of how we can use cell culture and animal models to go about connecting the genetic perturbations and biology, but also the environmental inferences through stress hormones, in this particular case, in an animal model. In parallel, Mary's team and the MVP GWAS have been started emerging, and not surprisingly, in a similar sample size, different loci were emerging. But when PredictScan and this type of ways are when I applied, again there was a very narrow list of genes that were predicted in the brain, again in the prefrontal cortex, to be associated with PTSD, and all of them were stronger in a locus in the genome that is called 17q2131. that includes one of the top gene and target of clinical trials PTSD called CRH receptor 1, which is the prime receptor of CRH in the brain. But at the same time, it's a very complex locus to find map because it has an inversion that is more frequent in the Europeans and also has many other important genes, including the tau gene of important neurodegeneration and other genes in other diseases. So we start forming the initial hypothesis, but still we are not there. And here is the Freeze 3 that was just released in April in Nature Genetics, in which we applied the methods again. And the cool part is that we have many genes. We can apply many more models, different type of statistics in the different models, but we can also start seeing that methods start agreeing in some location. One of them is the 17q2131. I'm using it as a common link in the presentation. It's also my favorite location in the genome, but it helps tell the story. But also another example is chromosome 2 that has a very strong and maybe the top loci of PTSD. Right now, the biological information we have been using cannot really say anything about the fine mapping of chromosome 2. So there is also still a lot of work to be done. But what was missing is like, can we go to real, all those predictions were primarily strong in the brain. So the question is, if you really go to the brain of PTSD, can you see changes in these pathways or other pathways? And these are the first 3 large-scale studies of transcriptomics of PTSD. Historically, those studies have been much delayed, primarily because it was difficult to collect PTSD brains in large scale. And there were some historical reasons that Joel Kleinman can explain the details of how he kind of solved that problem. But effectively, those studies start emerging in different sample size and also start agreeing on some topics and also not agreeing in other topics. One important observation is that the overlap of PTSD and MDD that is as high as 0.90 in GWAS, in genetic correlation analysis based on SNPs, that drops by a lot when you go to biological tissue, in this case in prefrontal cortex and amygdala. So looking at the relationship of the level of gene expression in particular brain regions, you see less of an overlap. So this means that you can distinguish them better. And you also start seeing some immune signaling being present, very strong statistically, but difficult to interpret of what it means and what is the origin of that immune signal, and the enrichment of interneuron markers as top differentially expressed genes across those studies. So the study that we wanted to do is to increase the scale of this investigation and to not only study gene expression, but study also epigenetics, proteomics, and genetics at the same time in a unified way. So this is the cover picture of the PsychoEncode phase 2 papers that came 2 weeks ago online, where the disease focus was not only PTSD and depression, there were many disease focuses. They were fundamental functional genomic calculations with a special focus on a cell type specific expression. And one of the papers was our analysis that I will describe to you. So the study design of our cohort, our project is called PTSD Brainomics Project, and it's comprised of three institutions. The big goal is to do a discovery replication analysis in 3 omic layers, transcriptomics, metabolomics, proteomics, but at the same time start to build up infrastructure to do large-scale single-cell transcriptomic studies initially. At the same time, we also analyzed from the lens of PTSD and MDD the data of UQ Biobank, and we also incorporated on the same type of biology that you see here what would be the fine mapped risk genes, because one of our main goals was to distinguish what are the disease processes compared to risk processes. The rationale of doing that is that when you have a diseased brain at death, all these things have happened. Basically, this individual was born with their genetics. They have been being exposed to pre-diagnosis stress factors, including childhood trauma. Eventually, they get a diagnosis. More stressful life events might happen, including treatments that are on top of the diagnosis, and eventually there is death, and possibly or frequently death by suicide. So when you get a tissue and you study it in postmortem tissue, it's very difficult to then disentangle all this complex trajectory. So here is the breakdown of the results. Something we saw here, and we quantify this with the number of distinct genes we detect with FDR signals, is that in both PTSD and MDD, top signal for discovery was at the genes and exon level of the prefrontal cortex, followed by, in the MDD case, again, RNA data from dentate gyrus of the hippocampus and central amygdala. But we saw that if you go to the details of these results, we saw that epigenetics, that was a main hypothesis for stress-related disorders, was relevant, but was not easy. It didn't have easily enough power as a function of how many CPG sites we measure. But we saw there was a difference between dentate gyrus being more relevant for PTSD and amygdala being more relevant for MDD in this analysis. Moving on, replication was a very important factor. We built up, the discovery cohort was approximately 231 samples, and we built and up to 114 replication samples. And we saw that in general, at the different levels of significance and at different levels of omic data, there was replication of signal with a correlation up to 0.6 when it comes to RNA data, while epigenetic data had lower levels of replication. And we saw some indication that PTSD was a more replicated signal than MDD, confirming our prior studies. So I covered that. And then one important topic we were interested in was what are the correlation structure between different brain regions, between different disorders, and different analysis you can do. So the original calculation was about the multi-region comparison. We're doing per omic layer. And we saw that was at best a low to moderate correlation we detected. And that the epigenetic data tend to have lower of those correlations, which is not a surprise given the importance of epigenetics in tissue and cell type specificity. Then moving on, we were interested if there are compounds or additional factors that are important for PTSD and depression that actually carry a big chunk of what we consider diagnosis-related signal. And we focus on death by suicide and childhood trauma. And we saw that largely in both disorders, indeed, childhood trauma and suicide are main factors explaining this signal, but not at the same degree in all omic layers, with the proteomic data having the lowest of those correlations. Another thing we observed that we would need much more papers to resolve is the possible sex differences that happen in the two disorders. We saw that with the overall signal, both the male and female PTSD specific analysis had moderate correlation with the primary signal, while this was not the case for the males in the MDD case. But we have to dive into comparing those sex-specific signals directly and do a more careful follow-up. And the last aspect we want to see is the comparison between the two disorders. And we saw that compared to the prior studies that I told you in the beginning, is that depending on the omic layer region of this biology we study, the correlation between PTSD and MDD can actually become low when you go, for example, in epigenetic data or some type of proteomic data, and can be high when you look at, for example, as the highest correlation we saw was in the prefrontal cortex gene expression. So by doing the investment of studying multi-omics at scale and in different brain regions, you can tear them apart in different degree. Also, this is, I think, significant given the fact that there is almost no PTSD brain that doesn't have some degree of depression in the record. So basically, it's very hard to, clinically even tear them apart. So it's important that biology can see the differences. And another thing is when you compare directly PTSD brains from MDD brains, that signal is not the same with the signal from when you compare them with the controls. Going forward, a very difficult type of investigation has to do with comparisons of pathways, because pathways compare omics with each other. It's very difficult to compare individual CPG sites that are at the level of letters of the DNA with whole gene expression or exon expression, splicing, and protein. So it's very difficult to do that comparison, and there are endless combinations. So we decided to first map them at common pathways, do pathway analysis across all this type of data, and then compare the signals that the pathways have. So we observed that in all those combinations, there are pathways like the immune signaling involvement that was common across gene expression, DNA methylation, and proteomics. But there were very interesting common signals in which the direction of expression and proteomics were opposite. That was something we did not expect. So apart from seeing low correlation, the existence in the top pathways of opposite correlation was a surprise to us. Additionally, we saw that there are many pathways that are supported only by DNA methylation. Most neuronal and synaptic signals were supported by DNA methylation and not by RNA. And there were also other unique signals in relation to metabolism that were mostly supported by proteins. Try to understand how this might be happening. One hypothesis is that it might be that the bulk signal you measure in association with the disease might be placed in different parts of the cortex. So we could do that only in the cortex because special transcriptomic data of all the regions we had did not exist, but for cortex they existed. So we say, okay, if we spatial register these signals of the RNA and protein, can we kind of tear them apart and maybe explain the differences this way? And we discovered that the RNA signal lies in it seems that it lies through this computational way. It seems that it lies in the upper layers of the cortex where you have the leptomeninges and other non-neuronal cell types, while the proteomic signal is deconvolved into more deep layers where it's more rich in neurons. So we had kind of an early hypothesis that might be a cell type specific issue in relation to proteomic and RNA data. Another thing we saw is that when you got the level of gene networks, that you have more power or more coordinated expression at the level of the RNA, and all those gene networks that were mostly associated with disease were immune related, and with a lot of stress regulation by glucocorticoid receptor or other modulators, that we calculated them to see that based on transcription factor binding sites of the target genes, and then we kind of imputed what we didn't do a ChIP-seq experiment, but we inferred it based on the targets. And you can see here GR supported by RNA data in MDD, but also by RNA and methylation data in PTSD. This is one example, but there were many combinations. There was a very strong effect also by estrogen receptor that we want to unpack later on for the sex difference part. Moving on, we also wanted to not to be biased in our analysis and follow a data-driven approach to this, not with our a priori ways of analyzing the data. And we were very interested in this type of approach that is called multi-omic factor analysis, MOFA, that is based on negative matrix factorization. And it's basically you load all the data you have. So you can imagine you have thousands of RNA features, thousands of methylation features, and thousands of proteomic features. You load them in MOFA, and MOFA discovers, based on some parameters that you have to optimize, it creates factors for you that could be either influenced, loaded primarily by one type of biology or by all biology. In this particular case, you can see factor 1, only methylation in MPFC is relevant for factor 1. But you can have factor 13 that all the omic layers and all brain regions play a role for factor 13. So you can also have factor 2 that is an RNA-only factor. So you can see all those combinations, but then we test it like, oh, these factors, are they related to anything? Can be, there is no phenotype file part of the MOFA analysis. It could be related to gender, related to age, related to many things. One very influential factor, this factor 13, turned out to be a multi-omic clock, data-driven multi-omic clock. We didn't run a regression against age. We found first factor 13 and then realized that is a multi-omic clock supported by all omics, all regions, and in some degree superior than the classic epigenetic clocks that we have been using. We had another factor, factor 14, that so the multi-omic factor 13 that had the age effect had also indications of age acceleration in both disorders. But the most strongly associated factor with disease was factor 14. But we would need a lot of work in like unpacking what is the biological structure explaining those data-driven factors. But we think that it was an interesting thing. And then we went to single-cell transcriptomics, so to test the cell type specificity of all these things. And we have already been working on one study in single-cell transcriptomics. Basically, this is the 10x genomics platform that relies on a microfluidic way of using oil drops and barcoding to isolate RNA signals per nucleus per sample. So suddenly, instead of having one smoothie of data point that has all the cells as a smoothie all together and one data matrix, you have all the cells separate, and for every subject you get 10,000 columns, and you get after doing a lot of filtering, and you get those type of maps that right now, especially for prefrontal cortex, are pretty reliable. You have a lot of references to compare for things created by Allen Brain Institute and others, can confirm if you're getting the same type of map, what cell types you get. In a gross way, you can see here the 8 major cell types of prefrontal cortex, and with the major markers that they are assigned to. But if you go deeper, you can go in the excitatory neurons, recognizing layers and subtypes of excitatory neurons. On the right side, you can see inhibitory neurons. You can distinguish based on those clusters that you can even figure out, embryonic origins of cell types, receptors that they express in their membrane. And this is all data-driven based on how the transcriptomic data is. But in the end of the day, there are many initiatives to study that biology, but you want to see, does this associate with disease? And when in an n equals 30 with 32, we did this. Our power was mostly at the neuron level for both disorders. We also saw that the correlation between the disorders drops even to a greater degree than bulk studies. But a remarkable thing that was a little bit mind-blowing to us is that genes of the 17q2131 locus from the GWAS was by far overrepresented in the top genes in this tiny study. So we are comparing a 1 million people study and an N equals 32 study, and you find these genes from that locus being differentially expressed. So that was like mind-blowing, that what explains that, right? And cannot be only luck, I guess. And then one other thing we saw is that these common upstream regulators that are more generic in when you analyze bulk data, now when you see them at the cell type level, you can see the cell types agreeing for some cell transcription factor, disagreeing, being in alignment, differences between disease. One clear transcription factor that is a focus in my lab, but it is like the receptor of the main stress hormone, cortisol, glucocorticoid receptor, have this pattern of difference between PTSD and depression. It has this opposing signal. It has this opposing signal in naive pathway analysis, but also in this upstream regulator analysis. But being the stress field, I have a tendency to be suspicious of this type of data, because when you run this type of analysis, you rely on a black box reference that you don't know. Is it the cancer cell line? Is it something crazy like a leukemia cell line that had treatment with glucocorticoids? What's going on? Can you believe if it's up or down, right? So we reanalyzed some data from a mouse model that did ChIP-seq and transcription to tease apart the two receptors for cortisol, glucocorticoid receptor and mineralocorticoid receptor, try to confirm and kind of like figure out the direction. And we saw that indeed it must have been GR upregulated as the signaling in PTSD, and less so MR, as also this prefrontal cortex and MR is primarily an hippocampus thing. And the direction was confirmed. So we say, okay, still I was doubtful, because again, this is a bioinformatic analysis. So then I'm like, we have to do the experiment. If not, we don't believe it. So we said, okay, we're going to do the most expensive pathway analysis. We're going to take control iPSCs, not related to PTSD, no polygenic score of PTSD on anything. We will culture them to the degree of maturity that we will see a functional glucocorticoid receptor. And if we go at that stage, we will expose them to the glucocorticoids, generate a data set. So this is what happens here. We see the translocation, we see the phosphorylation of the receptor, we get the RNA-seq out of this experiment, and then we say, is this signature opposite in direction in PTSD and depression? And this is what we did here. And you can see this remarkable change of direction. You have this positive RRHO, which is a similar type of thing with correlation. It's just that it is more threshold-free way of analyzing it. And you see this positive correlation with PTSD, opposite effect in MDD. And that was have been hypothesized for 40 years in PTSD research, but only in studies in blood. It was the first time you see it in brain. And another thing we observed is that at the same time, we have this strong PTSD, strong effect from the glucocorticoid receptor side, mediating the environmental influences, but we have this genetic influence, and we were trying to understand, is this 17q2131 so strong specifically for prefrontal cortex? So we analyzed the UK Biobank neuroimaging data, divided them into neuroimaging data that involve or not involve prefrontal cortex, used the GWAS as a basis, and then converted it to prefrontal focused gene expression associations, and saw that indeed 17q2131 has a bias to influence prefrontal cortex neuroimaging. And one element we had as a lack was like power. Like, is power basically influencing how much importance we put to neuronal gene expression and neuroimaging? And indeed, like in the science paper analysis, we actually aggregating much more data than what we had in the original study, and we see that indeed that the fact that we said that it was a neuronal only signal was biased from being underpowered in non-neuronal cell types, especially in MDD that we acquired a sample size of n equals 50 into the two groups. We saw a very strong signal in other cell types, primarily in astrocytes. But at the same time, we confirmed that in this higher sample size that the 70q2131 thing was true because we can replicate it in the higher sample size. And we confirmed the low correlation between the two disorders. And the last thing we did was, am I good with time?
No, you're fine. Yeah.
Yeah,
No,
yeah. The last thing we
yeah.
did was to say, okay, can this be also studied in blood? So we went to UK Biobank and we used the proxy definitions of PTSD and MDD that exist. And we did the proteomic association study to find in these 1,500 proteins the top associated with PTSD and MDD, and try to do the correlation between the effect sizes across the omic layers we had in the brain with what you get in the blood proteomics. The blood proteomics had the power of 54,000 people, but measured only 1,500 proteins. Our brain data were much more deep, but the discovered sample size was n equals 231. So the fact that we saw strong correlations, especially between brain RNA and blood proteomics, was mind-blowing to us. Of course, in the level of blood, we didn't have all the other omic layers, but in this level of interpretation, knowing that the deconvolution of the signal of RNA is primarily at the vasculature layers of the cortex, and that correlates mostly with what the blood proteomic signal has, we have a hypothesis that we have to test in future studies, is that there is at least partially a signal that comes from the periphery to the brain rather than from the brain to the periphery. But it remains to be tested. That was, to me, was not a surprise, because 10 years ago, I have done a study. Back then, single cell data did not exist, and we were kind of envisioning that this eventually will exist. But back then, we have done a study in rats that they have this innate fear for the smell of urine of a cat, but they saw an individual variation in the wild type background in that phenotype. They can be divided in the PTSD-like animals and the PTSD resilient animals. We have seen that the overlap of RNA data from amygdala and hippocampus and blood was low at the level of RNA individual gene expression, but if you would impute the proteins regulating that transcriptome, that overlap from 6% was all the way to 36%. So I was surprised to see this. Back then, we also had also the link with the glucocorticoid receptor as the main mediators of the common signal between the two. So I definitely want to follow up what does this mean in relation to what we observed in the human study. And one other topic that would be important in the new grant that we just got is that in this study, for the first time, we start doing the real causal analysis that so far we have been inferring, meaning that in people that we have genotypes, we have their molecular data, can we go in their brain and see that the SNP affects a certain gene and that gene predicts the diagnosis? In this sample size that I told you that is still limited, even if it's the biggest PTSD study in the brain, it's still limited. We were able to confirm these 6 genes having a proven causal mediation from SNP to expression to phenotype, but we want to do that much deeper. And one of those genes is this gene that was top gene in the sickle cell study, one of the top genes in the bulk, but also part of the same genomic locus, in which we also see some gene-environment interaction effects with childhood trauma. And this, I think, is still the beginning, because this type of analysis on the left side we cannot do yet at the level of cell type specific, while we know that there are some particular cell types that explain the GWAS signal more than others. And we most likely have to kind of go and doing this analysis at the level of cell type specific expression. This is the summary of like where our multi-omic, multi-region top genes are at this moment. And we envisioned a way of prioritizing biomarkers in the paper from what properties they have, and we tested our way of prioritizing if it gives you additional properties that you didn't go for, as a way of confirming that our prioritization was right. So we thought that, you know, a multi-regional characteristic or a multi-trait or a multi-omic property predicts strong signal in other things, in other type of data sets. But, you know, this was just as a proposition of like that we have to be thinking about what are the criteria of qualifying biomarkers for future studies. And I want to finish like with an open question is, like, can we use this type of genetically predicted molecular phenotypes and real biomarker data to go beyond the DSM? Basically, can we use this type of data to accomplish what that perspective, positioned? That basically, that if you have a heterogeneous group that gets diagnosis, if part of your diagnostic process was actual biology, you might have seen clusters of people that most likely have a biological component and maybe would respond to biological treatments in a less heterogeneous and responsive way. Very recently, we have tried to do this type of EHR type of work in data from the Grady Trauma Hospital, where we actually had genotype, very sparse biomarkers, including neuroimaging and blood work, and the genotype. For the genotype side, we created those imputed gene expression profiles, and we use this method developed in Manoli Skelly's lab that basically MixHR is trying to model in this type of diverse data where you have continuous values and categorical values and missingness to model at the same time non-missings at random, and the idea that some data are continuous, categorical, and they are different. And at the same time, we wanted to qualify. So basically, this is a clustering of variables. All the variables have different levels of missingness. You cluster the variables by observing the distribution that they have on their own, and then you create latent topics of variables that go together. And also you can, at the same time, model high values of one variable. Do they go with the same distribution with the low values or the high values of the other variable? So you create those latent topics, but you can also require in these iterations, do they associate with the main phenotype, in this case, PTSD? So we did that, and this model successfully trained this data, and then we asked those latent topics, can they predict subtypes of disease? And in this small study, what we observed is that we identified 3 subtypes of disease. One type of PTSD that has PTSD, depression, and childhood trauma, and one that is a PTSD-only subtype. And we also identified one resilient phenotype that doesn't have anything, and it's resilient in terms of phenotype. But at the same time, these subtypes are associated with all these biomarkers that we modeled. But I'm finishing that with kind of an open question. Can we improve this topic by these approaches. And my vision is that we can always start from the GWAS side, that is the ground truth, because it's like you're the time zero, design bulk studies and market studies that can help you up to a certain degree. And then more biological resolution we get, we can envision biological experiments and then basically back translate to the original population, see if what we discovered explains heterogeneity. And I want to help a lot of collaborators, and many people are in the room and in Zoom. Many collaborations with UCSD and my lab that is surviving this complexity, and you guys for listening. Please, we have 5 minutes for questions.
Nikos,
Yeah.
thank you. That was an extremely elegant and profound presentation, given how much integration across samples you've collected and how you've been able to masterfully translate genes to genetics to epigenetics to proteins all the way through. It's really impressive stuff. Along those lines, in our clinics, we don't have access to brains, but we have access to clinical samples, fairly rigid in terms of diagnoses. Fortunately, the computers force us to pick a diagnosis. We have access to blood samples. We can get bloods on practically everyone. There's not too many prohibitions around collecting blood, provided people give consent. We have severity indices, and we have a large integrated electronic health record that can tap into thousands and thousands and thousands of patient data. I guess my question is, can you take this approach without a few pieces and apply it broadly across a multiple array of diagnoses to really drive at the underlying mechanism that's mediating some of these variables?
Yeah, I mean, that's a very important question, and I think it will be very important in psychiatry to use all this type of data to improve from what you have now. I think my optimism would be yes. I think it will be helpful. We need to use the blood to get the appropriate omic data. We have ways of linking blood omics with brain omics with the help of reference panels and genetics. But the blood gives us the notion of time and moment that is advantageous, and postmodern brains will never have that. And I think we can iterate this, and then if we have biorepositories where we can go back to this blood and culture it or differentiate it into the right cell type, we can at least make a circle of where we kind of like start with this discovery, go back to the lab, test the hypothesis, maybe propose a drug, go back to the clinic, test the drug, do some clinical trial, test the drug, and iterate.
I see. So you can use the blood as a kind of a marker that you can then, traditionally we use animal models to see whether or not medications work, not always, but whether or not medications work through animal models and translate that to humans. But this is a way of being able to some extent complement that by getting into blood.
Yeah, I imagine that if from the proposed drug, you could potentially go back to the electronic record and test the hypothesis if the record supports the connection with, because many times it could be successful drugs, common drugs we have that we are not, without this stratification, we cannot see their effect. So we can go back in the record and confirm that. We can test in vitro many drugs on the cell types of interest, but I think the animal models, you will probably have to use them because you cannot easily go back to human to do a pharmacological clinical trial if you don't develop the right also animal model that figure out timing in an organism and more complex networks of cells. But yeah, I think all this could be a common glue using these approaches among I think electronic record, cell culture, and animal models.
So just, sorry, I don't mean to monopolize, but just in follow-up to that, have you done that with treatments? In other words, have you looked at treatments to see a kind of a, how treatments influence these? So, you know, look at someone in vivo over a time and then seeing what influence.
Yeah, in the Welcome Leap project that you guys are also part of, our project is trying to do that, trying to accomplish that in one empirical retrospective analysis of treatment as usual in McLean units, but also in an RCT study that we have. Thank you. But one thing is, like I was talking with Abraham before the talk, and he told me like some of these approaches I just presented, like the predict scan, genetic prediction of expression, he has now developed it in mouse models that could even give better precision.
Rat models.
Rat models, yeah. Rat models, yeah. Even better, rats are my
Yeah,
favorites, rodent, yeah. That gives, I'm biased because of my PhD.
Yeah.
I did for 5 years rats work, but this could give an extra link of like going at the genetic levels back to the animal models. Yeah. Any other question?
It might have been slide 27. Let's see if my memory is like,
Whoa.
I don't know.
Yeah.
But there was a thing you were doing a kind of a factor analysis like approach, and you told us a little bit about like factors 14 and 15 or so. And I was thinking to myself, typically when you do factor analysis, there's not much variance explained. If you know which slide I'm talking about, go ahead and go to it. There's typically not much variance explained by those distant factors.
17.
Right?
You
You know what I'm talking about?
1 second. Yeah, okay. Please go. 1 second. I
Yeah,
will,
on the screen though, yeah.
somehow. Okay. You talk about the upper right? Or something.
Yeah.
Yeah.
And so I just wonder, how do you think about that? And I think one of the very final slides. Right, exactly. And then I think this is 19. Sorry, my bad. And I think on one of your very final slides, you had another thing that looked to me like it might be a little bit of factor analysis. You're talking about disease subtype. And again, you were going way out. How do you think about that? Like these things are probably explaining only a tiny bit of variance when you get out there, right? If you did like a screen plot or whatever.
Actually, when we read the paper, there was not a very easy way of back calculating how they came up with the calculation of variance explained.
Okay.
We innovated a little bit, based on our understanding of the paper. But I think when you see the actual results of the MOFA, every single feature gets a minuscule weight to be loaded into to this factor.
Okay.
And we kind of like do a backward way to understand what percentage each of them does as an accumulation to the entire variance that factor 1, let's say, explains.
Okay, right. Yeah.
That is what we do.
Okay, because I mean, my initial thought was I looked at factors 1 and 2, which seemed really clear in a way. Maybe even factor 3 seems.
But you say, what is the overall?
Yeah, and I think if those are explaining a lot of the variance, which is often the case when you have ordered factors, then like let's focus there. It looks simpler anyway.
Yeah, I don't think that it works like in PCA. Factor number 1 explains more variance. No.
Okay, all right, perfect. Yeah.
Now, this tells you that methyl MPFC explains most of the variance of factor 1 in this population.
Yeah.
But it doesn't tell you that factor 1 explains more variance.
Most of the variance.
Like doing the regular.
Not why it's number 1.
No.
Yeah. Okay. All right. Perfect.
It's kind of arbitrary, I think.
Yeah. And then also, I think some of your proteomics stuff, like early on, you were showing us, and I don't remember the slide number, but it's several slides before this one. And it looked like protein from the UK Biobank, like the 50,000 blood from the omic thing or whatever, was not a very good predictor compared to most of the other things that you were looking at. And then later on, it seemed like the protein was working better. And I wasn't sure if I followed the reasons for that. Yeah, I think you might have just gone by it there.
So you say here, right? The proteomic data, these
This is
things
the one
are
I
here.
was thinking of, but there were several slides where it seemed like proteomic wasn't performing that
Yeah.
well. And then later in the talk, it seemed like it was, and I think it's the same proteomic data.
What happens here is
Yeah,
you have proteomics that way of quantifying what means explains, right? And basically, if you quantify it based on how many FDR signals you have within the proteomic data, yes. But then, in the gene set enrichment analysis, you load all the betas of the proteomic data, and it's trying to find with this GCA calculation, if there is an enrichment and a bias in the enrichment in positive or negative direction for a certain pathway as a whole to be. So that graph, using not the, I ranked the individual feature data, how many proteomics have, and they didn't have that many.
yeah.
But if they were coordinated, they could generate a pathway that has a pretty good p-value.
Okay.
For example, it has this nice red color, right?
Okay.
At the same time, while the
Yeah.
individual features themselves are not that strong,
Okay.
but as a composite,
Yeah.
for this particular top pathways, they are.
Okay.
So
That's sort of how
the level
both
of
those things are true.
yeah.
Yeah.
Now with the UQ Biobank, it's true that when you correlate a vector of effect sizes that in the proteomics is not as widely wide, doesn't have some extreme values. With the UQ Biobank, maybe RNA has a higher probability of showing you that nice correlation because it has a wider spread of effect sizes in the RNA brain data compared to. So in that sense, this is the only thing we could do. But we also tried in the paper to actually overlap top RNA, top protein, top methylation.
Right.
So there's more nuance to what I described if you go to the paper.
Okay.
Just like basically the short answer is at what level of comparison you make that conclusion. Also the sex difference of being stronger in MDD at that level of comparison, while we didn't fully explore sex differences. So it's kind of difficult to make a statement in a talk and actually be accurate completely, right?
Okay.
Yeah. So I guess it's not a definite definite win of RNA versus protein.
Right.
Yeah.
Okay.
Yeah.
Are there any internet people with questions? I wonder. I can't really see. Those you lose internet people at the end of the.
No, there's
Got
no.
one.
Yeah.
In
There's one, but I guess it's
Is that a question or?
No.
Great talk.
Yeah.
Thank you.
going back to your very first slide, you show us the HPA axis.
Yeah.
Presumably working on corticosteroids and glucocorticoid hormones and things. And if you look in the brain, there's a lot of differences in which neurons are expressing glucocorticoid receptors, and hippocampus is very high and other places like striatum are very low.
Yeah.
Do you see any signal for stress differences in terms of your neuronal signatures in the omic data that might map onto what's known about stress signaling in the brain?
That's a very deep question. Glucocorticoid receptor as a receptor is very abundant. Now we might know differences in levels of expression, but this doesn't necessarily mean that we know for sure the differences at the level of function. When we see the glucocorticoid distribution in this T-SNP map within cells of prefrontal cortex, we see wide distribution. We see, for example, for signaling, understanding signaling, you see FKBP5 that we know a lot of literature about the effects of FKBP5 mediating glucocorticoid signaling in neurons, but we see FKBP5 more strongly in non-neuronal cell types. And I think microglia being number one. So I think we don't know enough. We focused on the neuronal signal because it was the top differentially expressed signal. But then we saw that we didn't have the power and we will probably discover more genes. One of the genes that we got that maybe I did not show is that in the new volcano plot, no, that's slow waves, but that I think can make my point more clear. Sorry. So in here, we didn't have any glucocorticoid receptor gene per se, but the pathway analysis is my point that we made with the cell culture. But here, if you see here, you see FKBP5 being top differential expressed genes in 3 cell types, both neuronal cell types, the red and the, but also purple in oligodendrocytes. So I think probably what you said is right, like every brain region will be different and the cell type differences will be regional, and hippocampus will be a complex one because you will have also published before that, you know, CA1, CA2, CA3, and dentate have differences in that ratio and the involvement of FKBP5 and the other partners. So I think it will be a very complex topic. But the hope is if we can resolve these questions, a common drug like glucocorticoids could turn into a cell type specific treatment if we understand more. Now, how we would deliver it in a cell type specific way, it could be that we could develop some kind of an antibody based delivering of glucocorticoids in the cell type we would want and something like that. Because now when we give it generically, all this very wide distribution is against us, right? Also, if you have opposing effects and stuff like that. Yeah. I hope that.
Can I ask one more? That's kind of, so I know we're a little,
Yeah.
but this is kind of the other side of that question is like, to what extent do you think the amount of tissue that you have might bias your hypotheses? So you have 50,000 blood samples, but if you had 50,000 livers, would you be having a glucose metabolism hypothesis of PTSD?
Yeah, of course, we are biased from the data. Also, in all functional genomics data we have are so biased towards prefrontal cortex. And now there have been more recent studies, like maybe you have seen Mike Gandel's paper in Nature last year in autism. He makes a catalog of, I don't know how many cortical regions, and then top signal for autism is visual cortex. Right. While nobody has done any studies before on visual cortex, right? And I think in PTSD too, there are some neuroimaging studies that find this surprising, not traditionally studied brain regions. So I think similarly to the peripheral tissues, so I think we are biased to what the data we have. Yeah. Yeah. And if we can fix that, it would be amazing.
Early questions?
Thank you for active discussion.
Yeah, thank you.
We can see it or not?
Yeah.
Okay. Yeah, no.